So good evening. It's a real pleasure to visit Paris. This is my first international flight in 2.5 years. And believe me, to see people, to interact with people and have the opportunity to see Paris and talk about something I really love and I think is terribly important is a great pleasure. As Anton said, I'm going to cover the history of Analem's establishment and where the science came from and how the science ultimately is translated to patients, Akshay and our speakers will cover tonight. It's a very exciting story. It's a story of 20 years, and that's a rapid development for a new technology. And it's a story that I think has enormous potential to change healthcare around the world. But I want to start back at the early days of biotech because I think, well, for sure, Analem grew out of the early development of biotech in Cambridge, Massachusetts, an area called Kendall Square next to MIT. So I will start there and then move forward. So I'm a co-founder of Analem, chair of the Scientific Advisor Board, member of the board of directors, and I have been since the company was established. I want to think back to where and when we realized the nature of messenger RNA. Within the last 2 years, messenger RNA has become really part of almost everybody's life. Almost most of you in this room have been directly injected with messenger RNA. So this is another example of very fundamental early science that has become a major tool in pharmaceutical development. And what I show here is a model from Jacob-Monod. It was published in 1961, and there were a number of other developments in the same time, but there's no question that this model of the regulation of the lac operon was the model that many young molecular biologists who entered the field had as a vision of messenger RNA and regulation of gene expression. And what it shows you is 3 genes, Z, Y, and A, that those genes are transcribed into a messenger RNA, and that messenger RNA is translated into 3 different proteins, all of which are involved in the metabolism of that glucose. And at the left side of the slide, what you see is an operator, O, and that's where a protein that's called an oppressor will bind, and when the sugar is present, it comes off, allows the copying of that messenger RNA. And this model of Jacob-Monod, to explain their work, they received a Nobel Prize for, and it is the signature development 8 years after the discovery of the structure of Watson and Crick. Watson and Crick discovered the structure of DNA with Rosalind Franklin's data, and that told us what the chemical nature of inheritance was. And it took us 8 years to go to understanding how the information that is encoded in that DNA was then transferred into the cell by this process of transcription to make the messenger RNA and translation to decode it. And at the same time, we discovered the function of tRNA, the connection between RNA and protein, the nucleate code and the protein code, and we discovered the ribosome as being the translational machine. Now, this model dominated how we understood genes in cells until 1977. And in 1977, doing some experiments with a virus called adenovirus, it's now used to vaccinate people, but we were interested in how its genes were expressed. And what we discovered, and is shown here on this slide, and you see a lot of boxes, Some are white, some are dark. Those boxes are the pieces of the sequence of the gene, which is that center line, that are produced and transported to the cytoplasm to make the messenger RNA in your cells. So 99% of the genes in your cells, and there are only 20,000 of them. I'll tell you a nasty little secret. There are worms out there in that soil that have more genes than you do. But all the genes in your cells typically have the structure. The information for the protein is coded in black there. And what you see is those pieces of black have to be joined together by a process called splicing. And that then produces the message that goes to the cytoplasm that then is translated. So when Moderna or Pfizer produces a message, they're taking a message equivalent, a viral message, to this type of message and treating you with injection. Now, I don't want to leave you with an inferiority complex. So you're more complex than that worm is, because each of these genes can be spliced in different ways. So what you see in the middle there with that number 2 is that in some cells, 1 is spliced to 2 to 3, and you get one protein, and in other cells, you get a skip of that exon, and you get a different but related protein. So you have a level of complexity in how your genes are expressed that we really still are challenged to be able to determine. So we don't know what a given cell in your brain really has as a message in general, specific. In general, we know a gene is expressed, this gene, that gene, but we don't know the complexity of the splicing. Also in the 70s, we developed synthetic biology, and that is the ability to do recombinant DNA, DNA sequencing, and the chemical synthesis of DNA. So for the first time in the history of man, we could design genes. We could make genes, we could put genes in organisms, and we could have new properties of those, from those genes and the proteins they encode. And this raised the opportunity to take this science that had been developed over 25 more years to translate it into something that is useful for people. And that led me and Wally Gilbert at Harvard and 2 scientists, Charles Weissman and Bernard Mach from Switzerland, 2 from Germany, 2 from England, to establish a company called Biogen. Biogen was established in 1978. It's the 3rd biotech company organized, and it's the oldest freestanding biotech company, because Genentech was the first company organized in biotech, and it has been acquired by Roche. Cetus was the second. It was acquired many years ago. Biogen is a freestanding company. Biogen relocated to Cambridge, Massachusetts in 1980. It was originally organized in Geneva, Switzerland, and opened its headquarters in Cambridge, Cambridge, US, in 1980. Now, between 1980 and now, it was the first biotech company to locate in what we call Kendall Square. And what I'm showing you here is a diagram of the vicinity of MIT on the Charles River. So you see at the bottom of the Charles River, you see the red buildings. Those are buildings of MIT and the academic campus. And you see to the right, all the blue buildings and all this other markings. Everything blue there is a biotech. And there are about 50 to 100, and if you take in the small ones, 200 biotech companies in Kendall Square next to MIT. This all developed since 1978, and it is continuing to develop because this map of Cambridge or diagram is in 2018, and there is a large number of additional companies. Now, I want to point out a few. To the right, you see Biogen. Biogen has 6 buildings in this area. You see Alnylam up at the top, and we'll talk about Alnylam and its establishment in a few moments. You see Novartis has its major research institute 1.5 blocks from MIT. You see Sanofi Genzyme. Sanofi bought Genzyme, and ultimately it's become just Sanofi, about 3 blocks in a building there. And you see the Broad Institute, which is an institute that is jointly associated with both Harvard and MIT and independently endowed. And it was a Broad Institute and Eric Lander and the Human Genome Initiative that produced the first sequence, part of the first sequence, and an international collaboration for sequencing the human genome. So here in Cambridge, in 2002, 6 years before this, Alnylam was established down the street from Biogen, 3 blocks from the Broad Institute, 4 blocks from my office. Now, the Broad Institute was a major player in the sequencing of the first human genome, and that sequence has allowed an amazing advance in science. It's taken us from not knowing what genes are expressed in A cell versus B cell, the nature of mutations, the nature of changes in cancer that is driving our pharmaceutical development and science forward. It is a transformative advance in biological science. And the thing that struck me, and this was in 2001, the sequence was released, is it would be a wonderful advance if we had a way to take that genome sequence and design a therapeutic directly on the sequence of the genome. And that's what this process of RNA interference allows us to do. But before talking about that, let's talk about the advances of sequencing the genome. So the cost of the original first genome sequence was approximately several $1 billion. They started sometime in the late 1990s and completed the first sequence in about 2001. And what you see on this slide in the blue line is the rate at which the drop in the cost of sequencing a genome has decreased. And you see that in 2004, we're down into the 1,000. We're now approximately $200 to $300 to sequence a total genome of a patient or a tumor cell or otherwise. So this has made it available almost at the level of cost of an X-ray or an MRI or other procedures that are more frequently done in healthcare, and will increasingly be part of what healthcare is about in the data that comes from it. So what are we going to discover, or what have we discovered in looking at the human genome sequence? Now, this is from a review article in 2015 from a number of human geneticists who wrote a treatise on the nature of genetic disease in humans. And what it tells you in 2015, there are 3,000 million genes that are known when they have mutations to actually cause phenotype in man. And 8% of all live births are thought to have a genetic disorder that is identifiable by early adulthood in some phenotype. the estimated cost of each child that has a genetic disorder throughout their life for healthcare and probably loss of productivity is about $5 million. And the diagnostic rate in 2015 at the age of a child was about 11% of those who have genetic mutations, and about 24% as adults when those mutations become more overt phenotypes. Now that number is 6,000 Mendelian genes, and I suspect those diagnostic rates have increased by at least a factor of 2. So we have an emerging number of genetic variations in the population that are known to lead to disease, and effective treatment of those is something of incredible importance. So at the same time this was happening with the sequencing of the genome, there was a discovery of this new phenomena that we were not aware of before 1998. And that new phenomena is RNA interference. And it was discovered, as I will show you a picture of these two handsome young men, in the study of a worm, C. elegans, where they were trying to understand the function of a gene, and they made some RNA samples and injected it into the worm, and the RNA samples from the gene silenced the gene. But then they said, we don't understand how this works. And they then started exploring how it silenced the gene and came to the insight that it was a double-strand RNA, not that single-strand RNA that the message was in the first slide, but a double-strand RNA with the message and its complementary strand together. And they showed that it's silenced at a post-transcriptional stage by cleaving the message. And we now know that there are other processes that silence genes at the stage of translation, and I won't talk about any of that. And we know that in some organisms, such as the worm and the plants and many others, this is their innate immune system. This is their immune system. This is how they recognize foreign viruses and silence them. So Andy Fire, which is shown there to the right, and Craig Mello made this discovery in 1998. And Andy got his PhD with me at MIT. And we had kept in touch throughout his academic career. He's a friend, a great guy. He came to MIT as a 19-year-old freshman, 19-year-old student out of Berkeley with a hairdo like this. And just brilliant, an absolute brilliant man. And he worked on transcription in my lab and went off to Sidney Brenner and then became independent. And that's when he was collaborating with Craig Mello, this other young person here, and made the discovery of this double-strand RNA. They made that discovery in 1998, 2 years or 3 years before completion of the sequence. So here we had the discovery of the double-strand RNA is a silencing process called RNA interference, and we had the human genome sequence. A group of colleagues at MIT and myself decided that we would try to duplicate this process in a biochemical reaction so we could study the mechanism. And Tom Tuschl was a major player in this. Tom was a postdoc between myself and David Bartel, and Phil Zamore, another postdoc. And in the first experiment they designed, where they were using an extract, this is no cells, but a biochemical test tube, they put in double-strand RNA, and they found that they could see the same phenomena in a test tube that happened in the organism. And we knew that if we could get it in a test tube, we could analyze how it worked. And that led us, and Tom Tuschl particularly, when he went to Max Planck, to understand that this long RNA double-strand RNA, was cut into these little pieces of 21 nucleotides, and that's what you see there. 19 nucleotides of duplex. Each of those are representing a strand of RNA. You see those bars vertically, that means the two are complementary, like DNA is complementary. And it's got these little tails at the end. And that's called short or small interfering RNAs. But you can make 21 nucleotide RNA synthetically. It's very simple. It had been worked out long before. You can modify them. You have really total chemical control of that RNA structure. And I won't dwell on this, but I have to show one piece of data showing you up to the right, and this is the work of Tom Tushel that was published in 2000, 2001 in Nature, that you could use a small RNA to silence a gene in human cells. So what you see at the bottom is what is called a Herc stain, where you're staining the nuclei of these cells. What you see in the middle is the same cells stained with NUMA, which is another nuclear protein. And what you see at the top is that a large number of these cells are not expressing that lamina A and C proteins, which we're detecting with an antibody, because they have been treated with the siRNA, and that RNA has silenced that gene. In the next lane is a control, where it's another gene, and it's not silenced. So it's specific to the gene that they were active. So at the same time, in 2001, there was another major discovery, and that was all our cells are full of these small RNAs, which we didn't know. We have 1,000 or more genes who's expressed as these little RNAs called microRNAs. So in these studies by Tushel, Bartel, and Ambrose, published in Science and elsewhere, we discovered that these microRNA genes are common of all our cells, and they are critical in controlling activity of your cells. So we didn't know that. It's like, driving a car for 50 years and then suddenly discovering you have a transmission. It's absolutely central to the whole system, and we didn't know that it existed until 2001. So the process by which each of your cells make these microRNAs is shown here. And you see at the top the gene, DNA. It makes a little partially double-strand hairpin that's then cleaved into a smaller hairpin. It comes out of the nucleus. It encounters an enzyme called dicer that cuts it down to 21 nucleotides, just like that synthetic one we found. And it goes into a complex called argonaute or RISC. And then if it's totally complementary to a message, it'll cleave that message and silence it. If it is partially, it will inhibit the translation and cause degradation. So this is a microRNA pathway common of all your cells. And what that told us is that you have machinery in your cells to take small RNAs and pair them to message and to destroy or control that message. So that led us, I'll just show you this. The way these microRNAs usually work with message is they use a short sequence, not the total sequence. And what we had to do when we started developing siRNA is to control that it didn't do this, but it would only do the exact pairing. And I don't want to spend time, but this shows you that microRNAs control almost every pathway in the cell that contributes to cancer. So they have major critical roles in cells, and they are a part of all of our biological and physiological system. So this led us to the insight, that These small RNAs were part of the biology of many systems, plants, organisms, Drosophila and worms, and humans, and all vertebrate. And it became the scientific breakthrough of the year in 2002, and became highly acclaimed as a very important advance. And that led, this is Tom Tuschl sitting in a cold room in Germany, Max Planck. That led to the breakthrough, that establishment of Alnylam. So here in 2002, in Cambridge, scientific founders, myself, David Bartel, Tom Tuschl, Phil Zamor, and Paul Schimble, we recruited John Maganori, who was the CEO, and I first knew John at Biogen. So he was a scientist at Biogen and then became a leader at Millennium and then was recruited to Alnylam. And several of the first employees are people that had been recruited from Biogen into the company. The company went public in 2014 on offering on Wall Street. And in 2018, the company has 1,000 employees. I think it's 1,700 now. So it's a major biotech company. We've invested something on the order of $3.5 billion in the development of this technology before approval of the first product. And that comes out of the understanding that this technology is incredibly potently potential in affecting many diseases. So we have thought historically that therapeutics come of the type, a small molecule that inhibits the activity of a gene at the protein level, or an antibody or a protein therapeutic, insulin, an antibody to one of the HER2 treating breast, that interact with a protein, another protein target of a gene. Well, the target of Alnylam is to introduce the small RNA into cells, then have it loaded into that endogenous complex, the pathway I showed you the microRNAs use, be assembled into the RISC, and then to partially pair, or pair totally actually, to the messenger RNA from a given gene that we want to silence, and then lead to the cleavage of that gene. Oh, there should be another step here. Nope, it's not going to work. Lead to the cleavage of that messenger RNA, silencing the messenger RNA, and then being catalytic, doing it again and again and again. Because that system is present in every one of your cells and is used by the microRNAs that are regulating the genes of your cells. So Alnylam was established in 2002, and there were many challenges that Akshay is going to talk about, and then the therapeutic benefits of it, to effectively deliver these RNAs into cells. The RNA itself is hydrophilic. It lacks water. It doesn't pass through membranes. So to get it into cells, you have to solve the delivery problem. But Alnylam developed lipid nanoparticles that would do this. And the same lipid nanoparticles are now being used, same type, same chemistry, same optimization, are now being used for RNA vaccines. So you've all been injected with lipid nanoparticles if you've had an RNA vaccine. There's a GalNAc, another modification that allows delivery. We had to engineer stability, specificity. We had to avoid innate immune response. These are double-strand RNAs. Your system doesn't like double-strand RNA because it thinks it's a virus. and you have to be able to avoid that. And we're pushing forward to new frontiers, and the most exciting in my mind is the evidence that this type of technology could be effective in silencing a gene in the central nervous system. So I thank you for the opportunity to tell you this story. It's been an extraordinary experience being part of a team. It's mostly an alum who's taken this science and in 20 years created a new therapeutic modality. The last new therapeutic modality created was monoclonal antibodies, who had a platform potential as broad as this. So that happened well into the 1980s, 1990s. So this is a very important new frontier I am confident of for therapeutic development. So I think the floor is now turned over to Akshay, and I thank you for your attention.